Hello everyone. This lecture is about cancer vaccine. Okay, so usually we may think that vaccines are developed only against uh, infectious disease, but in fact, scientists are also trying to develop vaccine against cancer, and several vaccines have already been trialed, and several vaccines are on trial. Okay, currently there is no effective vaccines against cancer in the market, but some of the vaccines they are on trial on different phases, and some of those are really showing good results. Okay, so anyway, so the usual current treatment to treat cancer is chemotherapy, radiation, and surgery. So if we can add the vaccine, then it can be considered as a fourth modality of cancer treatment. Okay, so it will add a additional treatment strategy to treat cancer. Okay, and we can call it immunotherapy to treat cancer. Okay, so anyway, what is cancer? We all know. I mean, what happens in cancer cell? So usually in our body the cell proliferates, and after a certain cycle of proliferation they die. Okay, this is the usual case. But if and this process are controlled, I mean, how much proliferation will occur and when the cell will die, these things are regulated in our body. But if this regulation somehow fails, I mean the cell begin to proliferate for indefinite time, or if the mechanism by which cell die is damaged. Okay, if that mechanism don't work no longer, in that case what happens, as the cells begin to proliferate without any control, tumor forms and eventually cancer can develop. Okay, so that means the cancer cells, they are different than the normal cells. Okay, so what happens to the cancer cells? This kind of things: immortalization. Okay, in the definite proliferative lifespan, I mean the cells just continue to proliferate. Transformation. Okay, loss of response to normal regulators of cell growth. Okay, then the regulation system of cell growth is gone. Okay, because the cells no longer responds to those regulators. And metastasis. Okay, so the cell, I mean the cancer cell, they have the ability to leave a tumor and to invade other cells or tissues at another location. Actually, this happens in not in the early stage of cancer. This happens in the uh, third or fourth stage of the cancers, when the cancer, I mean, at the beginning, tumor forms. Okay, and it I mean it remain localized, but then when the cancer progress, some cells they leave that tumor and spread or invade into other cells or tissues. That means the cancer then spread throughout the body. This is called metastasis. Okay, so these are the things that happens to cancer cell. Okay, and as cancer cells are different, and in fact cancer cells are different than normal cells. So this slide shows some of the basic differences, such as cancer cell has a smaller cytoplasm compared to the normal cell. They have multiple nuclei, multiple and large nucleoli, coarse chromatin, etc. Okay, so these are some basic differences. Okay, but in fact the cancer cells, they also differ in many other ways, such as in the cell surface of the cancer cells, many different, I mean cancer cells, they express many different proteins, so that's why they become cancer cell. And also the cell surface of the cancer cell, they also express different kind of proteins. Okay, proteins or antigens, macromolecules. Okay, so when we want to develop a vaccine, okay, so the best approach would be to use those abnormal uh, antigens expressed in the cancer cell. Okay, okay, so the cancer vaccines, which which are under developmental process, they can be uh, classified into two broad group. One is preventive type vaccine or prophylactic vaccines, and another is treatment or therapeutic vaccines. Okay, the preventive vaccines, they work like the vaccines that we use to prevent infectious disease. The thing is, the hypothesis is, if we take the preventive vaccines, they will induce antibody in our body, and that antibody will protect us from getting cancer. Okay, but the treatment vaccine is, suppose a person already has the cancer, so these vaccines will cure that person. Okay. Okay, and there are five different types of cancer vaccines under development process. Okay, so what are those five types? Antigen vaccines. In this case, the tumor specific antigens are used as vaccine. Whole cell vaccines, that is the killed cancer cells, can be used as vaccine. Dendritic cell vaccines, so the patient's dendritic cells are used to develop dendritic cell vaccines. Idiotype vaccines. Okay, so in this case, idiotype antibodies are used as a antigen, that means are used as vaccine. Okay, and finally DNA vaccines. Okay, so okay, now in the later slides, I will discuss more about this vaccine. Okay, so first, dendritic cell vaccines. So in this approach, the patient's dendritic cells are, the dendritic cells are, they are isolated from a cancer patient, and these dendritic cells are used to stimulate immune response in the patient. So how actually it is done? Okay, so before talking about the approach of dendritic cell vaccine, first let's see what dendritic cell is. So the dendritic cell is a rare immune cell which recognize, process and presents foreign antigens to the T cells of the immune system. Okay, and the dendritic cells, they constantly roam around in the body and function as APC, that is antigen presenting cells. Okay, so what is the approach to develop dendritic cells? So first, we have to select the antigen. That means we have to select the tumor antigen which we want to use as a vaccine. Okay, so that means we want to use, select the antigen against which we want to develop the immune response. Okay, so first identify that antigen and link that antigen to a cytokine. Okay, that means add a cytokine with that antigen. Okay, now we have the antigen. Then we have to isolate the dendritic cell from the cancer patient, okay, or the tumor from the cancer patient, and then those isolated dendritic cells are cultured in the lab to make more copies of the dendritic cells to increase their number. Okay, so then we have to feed the dendritic cells with the antigens, okay, with our processed antigens. So if the dendritic cells, they get the antigen, they get those tumor antigen, as dendritic cells are antigen presenting cells, they will engulf the antigen, process it, and then present it in their cell surface. Okay, okay, so now we have the dendritic cell which is presenting the tumor antigen. Okay, so then this dendritic cell have to re-inject into that patient. Okay, and once that dendritic cells is inside the body of the patient, as it is already presenting the antigen, cancer antigen in its surface, it will attract the T cell and activate it. And now these activated T cells will attack and kill the patient's cancer cells. Okay, so this is the approach of dendritic cell vaccine for cancer. Okay, so in the box I have uploaded this book. Okay, vaccine design. It's actually a nice design. Okay, so if you check the chapter 43 of this book, you will find different. You will learn. You find different. Find information about different cancer vaccine which are which are in the development process. Okay, so I will suggest that you please have a look there, and also please have this book in your collection. You will find the different methods and protocols for vaccine design. Okay, this is actually a nice book to have in collection. Okay, actually this slide I just took from that cell. So this one, Provenge, it's a dendritic cell based cancer vaccine. Okay, and this vaccine has been developed uh, to treat prostate cancer. Okay, and the antigen used to develop this cancer was PAP. Okay, prostatic acid phosphatase. So this uh, I mean this this antigen, which is a protein, is expressed by many prostate many prostate cancer cells. Okay, so that why it was chosen to develop cancer by using the dendritic cell cancer vaccine approach. It was uh, tri- trial was done with this vaccine, and unfortunately the result was not promising. Okay, so this vaccine failed to cure the patient, rather it was just able to increase the lifespan of the patient for 4 months. Okay, anyway it is still yet have the FDA approval to treat to be used to treat prostate cancer patient. Okay. Okay, so this was about dendritic cell vaccines. Then another type, anti-idiotype vaccines. Okay, so what is the idea for this type of vaccines? So there are some antibodies which can act as antigens and trigger the immune response. So these antibodies are called idiotype antibodies. Okay, so if you want to get idea about anti-idiotype vaccines, first we need to understand what idiotype antibodies are. Okay, so what is idiotype antibody? To understand that, let's try to understand this idiotype network. Okay, so suppose this green one, okay, this triangular green one, it is a cancer antigen. Okay, so if we get in a, if uh, suppose a person have cancer and he is expressing this antigen, so what will happen? The body, the body, that person's body will develop antibody against that antigen. So this is that antibody. Now if this antibody is collected from that person and then injected into another host, not human, say another host means say the into the mouse. Okay, so what will happen? The mouse will recognize it as a foreign particle and will induce antibody against this antibody. Okay, so this induced antibody in the mouse, it has a portion. It's an antibody. That means it have a definitely it is a three-dimensional structure, right? And portion of this three-dimensional structure will actually represent the similar structure to that antigen. That means portion of this antibody will be similar, structurally similar to the antigen. Okay, so this is called a idiotype, idiotypic antibody. Okay, and this can be used as a vaccine. Why? So now if we isolate or collect this antibody from the mice body and then inject it into the that person's body, that okay, and then if we then inject this into this anti-idiotype antibody into that person's body, then as this antibody have a portion which represents the structure of the antigen, that means it acts as a epitope and as a antigen. So now that person will induce antibody against this antibody, and theoretically this induced antibody can react with the original antigen and neutralize it. Okay, so this is the approach for developing anti-idiotype vaccine. Okay, and if you search the Google, I mean, if you Google it and look for anti-idiotype vaccine in the Wikipedia, actually their page, they have uh, briefly write how the idiotype vaccine are developed. Actually, the same thing I have just explained, so you can give a look. Okay, and now the question is how the idiotype vaccine work. Okay, so I mean, what kind of immune response the idiotypic vaccine induce in the host body? Say, okay, say you have the idiotypic vaccine. That means you have that idiotype antibody. Now, if this antibody is injected into the host body, okay, so what will happen? Now, the host body has different type of immune cells, right? They have B cell, they have antigen presenting cells. Okay, so now some of the B cell, they will be able to recognize the recognize that idiotype antibody. Okay, so once this B cell recognize this idiotype antibody, they will bind with them and they will be get activated and become plasma cell and will produce antibodies, and that antibodies will now interact with the host cancer cells and kill that cancer cells. Okay, and this killing or destruction or lysis of the host cancer cells, it can happen in different ways, such as some antibodies may directly I mean the antibodies may directly interact with the host cell and host cancer cell and cause it and induce the signal which are responsible for apoptosis. Apoptosis means cell death. Okay, so some antibody, they will interact with the host cell and through the signal transaction induce the apoptotic signals and cause the cell lysis or cell destruction. Okay, and some antibodies, they will lyse the host cancer cell through with the help of complement system. Okay, some will induce cell mediated killing of the host cancers. Okay, so anyway, so this response will occur if the when the B cell of the host will recognize the anti-idiotypic cell. Okay, this is one way. Okay, so another way is the host cell has antigen presenting cell, so it will interact with the idiotypic antibody and then it will engulf it, process it and present it. Then once presented, the T cell will I mean it will attract the T cell. Okay, so now depending on the T cell, okay, so that means which T cell are attracted to the antigen presenting cells, the response can be different. So for example, is CD8 T cell, they get attracted, they will bind with that antigen presenting T cell antigen presenting cells and they will become activated. Okay, cytotoxic and become cytotoxic T cell and they will attack the tumor cell and kill it. Okay, and on the other hand, if CD4 T cell are attracted by the APC cell, okay, so they will release cytokines, which cytokines will activate the B cells and the B cells will become plasma cells and produce antibody against the idiotypic antibody. That means this antibody are able to kill the original antigen of the host cell. Okay, so these are the ways or mechanism by which the idiotype vaccine work. Okay. Okay, so there are already idiotypic vaccine under development. Okay, so if you Google Google it, so you will find this vaccine, Racotumomab. Okay, so this vaccine was is originally developed by Cuba, and they are under trial to check their efficiency against lung cancer, breast cancer, and some neuro uh, neuroectodermal pediatric tumors. Okay, so this is an idiotypic vaccine. Okay, I mean I took this photo from this paper. Okay, so I have also uploaded this article in the box. So please download it and try to have a look. Okay, so that's all for this part of cancer vaccine. In the next part, we'll learn about other types of cancer vaccine. Okay, thank you for your patience.